All right, in this section, we're going to discuss the evolution of photosynthesis, starting with cyclic electron transport. As simple prokaryotes continued to evolve, the systems of ATP synthesis and electron transport evolved more efficient and more complex systems. In cyclic electron transport, electrons are returned to the pigments after proton transport. So remember from the previous section, we talked about how the movement of those electrons in the photosystem provided the motive force to create that proton gradient to then generate ATP. In cyclic electron transport, after that electron moved, it returned back to the pigment in the photosystem, which you can see here. So again, remember, here's the photosystem, electron moves. You can see the facilitated diffusion of this hydrogen ion, this proton, creating this rotation to generate the ATP again. But the point here is to say, as these cells evolved, these electrons are then transported back or returned back to the pigments in the photosystem. All right, so look what's added to this process. We have this cytochrome added to the process to enable the transport of electrons from the photosystem to the specialized protein that functions as an efficient transporter of hydrogen ions across the membrane. So you see these hydrogen ions across the membrane so that we have a high concentration inside, a lower concentration outside. Remember, that enables the facilitated diffusion across the membrane at the ATP synthase, driving that conversion of ADP to ATP. Though this system was highly advanced at the time, evolution led to changes in parts of the system that made it more efficient. One of the major problems is that the ATP is being released outside the membrane. So this ATP synthase process is happening outside the membrane. That means the cell must then uptake the ATP from its surroundings. It must move it inside to be able to power the cellular work. Although ATP is a good molecule for energy management, it cannot be used for storage of energy because its reactivity causes it to break down quickly. So ATP-producing organisms can only survive when sunlight is available or if they flood their environment with ATP. Prokaryotes evolved in other areas in addition to ATP production, including energy storage, symbiosis, endosymbiosis, and compartmentalization. First, energy storage. Glucose is produced via photosynthesis using ATP and carbon dioxide, filling the need for long-term energy storage. If energy were money, ATP would be a quarter, enough money to operate a parking meter or washing machine. Glucose would be a $10 bill, much easier to carry around in your wallet, but too large to do the actual work of paying for parking or washing. This marks the beginning of sugar and carbohydrates on Earth. Next, symbiosis. Bacterial cells of different species tightly associate with one another and use each other's products and reactants. So you can see one providing ATP and the other providing glucose back to the first in this symbiotic relationship. Endosymbiosis. A key moment in the evolution of life is the point at which prokaryotic cells begin to combine into more complex eukaryotic cells. Several species of prokaryote, each with a separate function, joined together to form one cell capable of multiple functions. This is known as endosymbiosis. Evidence supports the endosymbiotic origins of both chloroplasts and mitochondria. The diagram shows an ancestral host cell absorbing anaerobic bacterium and a cyanobacterium over a long period of evolution. You can see the aerobic bacterium, the cyanobacterium. The aerobic bacterium became the mitochondrion, to be discussed in cellular respiration, and the cyanobacterium evolved into the chloroplast, which will be our next topic. Both chloroplasts and mitochondria have their own DNA, separate from the nuclear DNA. That's, of course, because they ancestrally were once separate cells, cyanobacterium or aerobic bacterium. This DNA is circular, resembling that of prokaryotes due to their prokaryotic origin. As the smaller cells, those cyanobacterium or aerobic bacterium, known as plastids, as those plastids are engulfed by the larger cells, they become doubly wrapped in membranes, which you can see indicated by this arrow here. Both chloroplasts and mitochondria possess double membranes, indicating this ancestral evolutionary origin. Endosymbiosis increases the metabolic ability of cells that descend from this process. Finally, compartmentalization. Some cells begin to get infoldings and more complex systems of membranes. This produces isolated pockets that can house separate chemical reactions. Cells can perform multiple reactions without interference from each other.